All right, in this short video, I just want to touch on some of the more important, well, maybe not more important, but definitely some of the cells which are talked about more in the immune system and highlight some of their major features. There's a lot that could be said on this, so I won't go over everything, but talking about some of the basics. And then I have a lot of scanning electron microscopy images here. I think they're really cool. You can spend a lot of time looking at them, and I'll touch on those a little more when we come across them. So leukocytes, term which will characterize all white blood cells. You hear the term white blood cells, and it's a very general term. And all that means is you could be talking about any one of these cells presented over here on the right, including more. This is, only a small fraction of what we consider white blood cells. As you can see here, they're all derived from this common cell up here, and this sort of looks like those trees of I'm not that sort of resembles that. Have a common pluripotent hematopoietic stem cell, and this is a stem cell which, when given the right sort of cues, will either fall down this lineage, which is the common myeloid progenitor. We talked about that last time. And this side over here is the common lymphoid progenitor, which gives rise to the lymphocytes. And so this talks about these two major lineages then. Myeloid cells, this houses all of the, actually houses where you make your red blood cells, your erythrocytes, the platelets, and then some of the ones we talked about last time, which are the macrophage, the neutrophil, the dendritic cell. These are all on this side of this developmental tree. And these other ones, like the basophils, eosinophils, I don't see mast cells on here unless I'm missing it, but mast cells would also be on here. Oh, there it is. These are known to play important roles during allergic responses, asthma, and then also it's thought that their main function is to deal with parasites, although that's a still hotly debated and researched area of the field. So this is what we call the myeloid cells. Over here, we have the lymphoid cells, with one interesting exception. This natural killer cell, it is an innate cell, and it winds up present with many of these cells before an adaptive immune response. However, it is lymphoid derived. And so over here, we have the T cells and the B cells. At the very end of that B cell differentiation is the plasma cell, which I've mentioned in the past. And this is what secretes antibody. So just having the B cell alone, while it does have some surface Ig, some surface antibody, it doesn't secrete it. It needs help from this T cell to then become a plasma antibody secreting cell. And so that's just kind of an overview of how these cells are broken up and characterized. Another way of thinking about this is we consider a subset of these cells called phagocytes. And so these are, to phagocytose is to engulf. And so these are cells that can engulf pathogens. We think of these as definitely the macrophages, definitely the neutrophils. The dendritic cell has some phagocytic capabilities as well. And then we also have granulocytes. Granulocytes are, as their name implies, cells which have cytoplasmic granules. And we can actually see them visually from here. We have basophils, neutrophils, eosinophils. These all have cytoplasmic granules, which they will dump these toxic granules onto pathogens. I believe in macrophages may, but they're not grouped into that. So just 4 or 5 slides about, I think, what are maybe the most important cells to know about. Touching again on the neutrophils, they are a phagocyte, they're a granulocyte. These are just ways to characterize them. They're found in the blood most abundantly, and they're not typically circulating in a tissue unless there is an infection, and that is because these are highly destructive cells. They have reactive oxygen species in their granules, they have hydrolytic enzymes, antimicrobial peptides, and they can even shoot out their own genomic material in this sort of like net, which will trap pathogens. They're really interesting, they're really important, and they're pretty destructive. They're the first responders to an infection, and therefore they're they don't really live that long. They do what they need to do, and they create a lot of damage, hopefully to the pathogen more than the host, because it is non-specific, and then they die off. A major component of what we call pus, I'm sure everybody's familiar with pus, is the neutrophil. So that white, pussy, well, that's not a word, pyogenic, purulent material is primarily neutrophils. And I mentioned they engulf pathogens and they have these chemical weapons. Sorry I didn't have that up sooner, but this is looks like a CGI rendering of what a neutrophil is. It has this multi-nucleated, well, it's multi-lobulated nucleus. So it is one continuous nucleus just with these lobes in it. And for that reason, they're sometimes called polymorphonuclear cells. So they have this polymorphonucleus nucleus here. Another important cell, which was that first yellow cell from the last video that sort of kicked off the entire process here, was the macrophage. And this is a scanning electron microscopy image. And these are phagocytes, so again, they engulf pathogens. These are resident in tissues. They're always in the tissues, they're underlying the tissues, as well as circulating through the blood in an immature form. They have these PRRs, which are pattern recognition receptors, and these are really aimed at identifying microbes through some of those conserved molecular patterns that microbes have and we don't. When that happens, they secrete cytokines, those purple cytokines from the last video, and that will draw out and recruit other cells into the infected area. They also play really important roles in wound healing and turning down some of the inflammatory processes which occur as a result of infection. They clear away dead cell debris like those neutrophils, and they can also present antigen to other cells through their major histocompatibility complex, which I talked about in the last video. They're long-lived cell types, so they can persist for weeks, months. I don't know about years, but definitely weeks and months. Yeah, and so that's an important feature about them. They do get to sites of infection pretty rapidly, not as quickly as the neutrophils though, unless they're already there. You know, they happen to be at a good spot where the microbe enters. So moving on to that dendritic cell, another scanning electron microscopy image. This is really, really neat looking picture. It's false colored. So we add these colors after the image is taken, but you can see it's got this really amorphous shape to it over here, and that is just a distinctive feature of these cell types. They have these arms, these filopodia, which will reach out and interface with microbes or other immune cells. These cells can engulf material, and when they do, they take it to the regional lymph node where they will present it to immature T cells and B cells. And what they're trying to do is find a lymphocyte there which recognizes the peptide it's presenting on its MHC. And that allows the dendritic cell to say, I found something out in the periphery and I'm holding it in which you know is me, the dendritic cell. And therefore, if you recognize this, we need to know that because that cell will be called out into the tissues to deal with the infection. They secrete a lot of cytokines. They can direct the development of other cells through those cytokines. Just the presence of a protein cytokine can really change the developmental fate of a cell, and these are really important for that. They're long-lived, and I mentioned that they are the interface between the innate and adaptive immune system. These cells are part of the innate immune system, and they recruit the adaptive immune system. And they are the only cells which can actively that can activate T lymphocytes. And so when a T lymphocyte is fully activated, it is licensed to go out and kill without some of the checks and barriers typically needed. For example, like a CD8 positive T cell. Once it's activated, it no longer needs to look for some of these inhibitory cues it would normally get. And so the dendritic cell is really important for licensing that cell to do that, for allowing it to do that. So the last two cells here that I want to mention were the T lymphocytes and the B lymphocytes, the parts of the adaptive immune system. Again, another scanning electron image of a T cell, and this is interfacing with, I believe the caption said it was a cancer tumor cell. These are really the superheroes of the immune system. These are what we call effector cells, just like the B cells would be. And they live for a long period of time as memory cells once they've been activated. They can directly kill cells, like CD8 positive T cells will directly kill host cells that are virally infected. But they can also secrete cytokines and direct the development of other portions of the immune system. They're broken up into two major lineages, the CD4 and CD8 positive, meaning positive just meaning it has that thing. So this is CD4 positive, meaning it has CD4. And these are important proteins that are on the surface of the cell, which interact with the major histocompatibility complex, the MHC complex. And so there are specific two types of MHCs. This is specific to one type, and this is specific to another. I don't want to get into the details, just want to mention that. I think the really cool thing about T cells is there's a lot of them. There's many subtypes, and they're all specifically tailored to deal with a different type of pathogen, whether that's intracellular, whether it's an extracellular bacterium, whether it's fungi. These all have different roles in that. They also play a major role in tolerizing the host to itself. And so they prevent autoimmunity because of that. Oh yeah, and so they're highly specific. I've mentioned that probably a number of times, just like the B cell is highly specific. This is another SEM of a B cell now. When this cell fully differentiates through those cytokine cues and the help of a T cell, it will turn into a plasma cell. And I've said before, the plasma cells secrete antibody. That is their main function. They will take up residence in a bone marrow. Others, some of the short-lived plasma cells will be in the tissues, but these just are antibody factories. They pump out antibodies. That is their main job. With the help of T cells, they can tailor that response, whatever that antibody is. And there's a lot of details, so I'm not going to go into it here, but there's multiple types of antibodies, and the B cell and the T cell interacting can make that response even better to find the exact right antibody to fit the microbe that it's trying to kill. And then just wanted to show two last pictures I thought were really cool. So this is, in the blue, you're seeing a dendritic cell. This is another scanning electron microscopy image. And then these are T cells, which are talking to that dendritic cell. And what they're doing is you're seeing if it's carrying any antigen that they recognize, because then they can activate and they can go out into the tissue and kill that thing, which is their favorite thing to do. So this is what happens inside of lymph nodes. And this is just another example of that. I think that these were T cells, and it said that this was a tumor. But this is just T cells. You're seeing a scanning electron image that's been colored, but T cells are actively moving and migrating towards this thing, which they recognize something is wrong with it. And so they want to do what they can to kill it. All right, and so with that said, I'll leave it here. I think there's a lot of details, again, that have been glossed over, but this is going to give you at least some sort of general background on what we mean when we talk about white blood cells.